Hi, I'm Ben Scott. I'm a new professor in psychological and brain sciences. And I started my lab in September of 2018, and we're focused on understanding the neural circuitry that underlies decision making. We think this is an important question for several reasons. The first is that a number of neuropsychiatric disorders, such as addiction, schizophrenia, autism, can be viewed as disruptions of the decision-making process. And the second is a paradox, which is that agency, which is so tied to our feeling of our, who we are as humans, is also at odds with the deterministic universe in which we live. And so we think by addressing this question, we can gain traction on both practical and also philosophical issues. Our approach is to train rats to play computer games. These games are not for entertainment purposes. They're inspired by tasks from the human cognitive literature to assess things like working memory and other aspects of cognition. We train them to perform these tasks in a high-throughput automated training facility. These are systems that operate 24 hours a day, 7 days a week, and allow for both the training of large numbers, in some cases hundreds of animals per day, as well as the generation of very large-scale data sets. So here's an example of a data set I collected using this system as a postdoc. We trained 7 rats to perform over 500,000 trials, and that data set allows us to fit quantitative models of decision-making to test hypotheses about why animals are choosing one course of action over another. However, we're also very interested in understanding the neural circuits that underlie these processes, and here we faced a challenge, and a challenge that most of the field faces, which is how we integrate these automated behavioral training facilities with advanced optical imaging technologies. One of our solutions is indicated here. We call this voluntary head restraint. So this is an apparatus that trains rats to basically operate a 2-photon laser scanning microscope. So this rat, we use the high throughput training facility to reward the rat for inserting its head under this 2-photon microscope, at which point it starts to play the computer game, and we can record large-scale population dynamics with calcium sensors, much of what Shui Han was talking about earlier, using this type of apparatus. A second technology that we've more recently developed are wearable head-mounted microscopes that have very large fields of view to record the coordination of brain activities, the coordination of multiple brain regions simultaneously during behavior. So here's a schematic of the rat brain. On the left, I'm showing the field of view of typical or existing technologies for the field of view of existing head-mounted microscopes. And on the right is the field of view we were able to achieve with our microscope. We call it C-scope, for cortical macroscope, because it enables a roughly 8 millimeter diameter field of view to be imaged while implanted on the animal's head. And on the right, you see 2 rats, one of which is a cage mate and one of which is surgically implanted with C-scope. We're imaging this during an open field navigation. And here on the right, you can see the output of this microscope. These brain regions, this is the visual cortex, the parietal cortex, the somatosensory cortex, and the motor cortex. So we can record across multiple brain regions as the animal's making decisions and image the flow of activity using this type of approach. Thank you.